So the outline of my presentation. So we'll give you a little and quick presentation of our scientific park. And as I mentioned, I will mention two projects. One is called Macrosys on animal genomics, and the second one is called Rice Immunity as on plant genomics. And I will give you also some future prospects of the work that I will be carrying out, particularly when, with Vittorio here in collaboration. So I call it our PTP cluster a small area because, you know, being here, it's much wider, you know, the kind of campus you have here. We started off our activities around 6 years ago. And as you can see, it's a combination, okay, of university. So we have the vet faculty and the agronomy faculty from Milan, and of other institutes, research institutes, and some other activities which are concentrated here. So this one is the building where all the research activities are carried out. It's 9,000 square meters, very colorful, as you can see. And so this one is a close-up. So as a description, we are the center of excellence of the Lombardy region for the biotech applied to the agri-food sector. So all of us have something to do either with agriculture or food-related aspects. So as you might know, Lodi is located south of Milan, and as I mentioned, we are tightly linked to university. So internally, we have a research center where also my activities are carried out, and a bioincubator, which is a place like here in area where we host, different spin-off from university or small medium enterprises. And we also created a business park, which is not active yet, but we hope some of these small enterprises will become successful and then will develop their own business inside a business park. So our general mission is to develop innovative solutions on the agri-food sector to support Italy, even if this moment is very difficult also for research. And we also do a lot of technology transfer to small and medium enterprises. So I mentioned the business incubator. So some of my work is also to identify new ideas, okay, and then develop business plans, and then verify market applicability or market prospects, do some economic feasibility, and then try to help, building up new codes. Presently, we have 15 new codes. You can look at our website, and you might find some are interesting because they're actually carrying out a link between the food and the nutrition. Okay, so this one is a link that is becoming stronger and stronger, and you might know, maybe some of you are already working on this in this field, which is called nutrigenomics and nutrigenetics, where, as you might know, we study the relationship with genetic variants to habits or diseases. Okay, we also have an international cooperation aspect. So we recently hired a scientist working in this field, and so we developed a number of research projects with South countries, mainly in the food safety area, because as you know, food safety is a very important issue. Other projects are more on rural development, and so we work mainly with non-governmental organizations. And we also carry out in much wider scale here junior scientist research training. So the general mission in research, what we aim to, it's to carry out high quality science. Of course, high quality science in the animal and plant sector. In an international context, addressing basic and applied research. So I guess from what I know of ICGB, we try to translate lots of our basic science research activities into applied aspects. So it could be patents, could be diagnostic kits, could be all sorts of other. I think you also have components more towards applications or towards market or maybe selling new ideas or know-how to maybe pharmaceutical companies. Okay. And so internally, we have around 60 researchers, and we're divided in two areas, animal science, and we have 3 different, what we call sections, and plant science, where we have a general plant genomics. And I also created this unit when I joined around 5 years ago. Alongside the research labs, who are mainly focusing on basic science research, we have these 4 platforms. So those are central platforms supporting all the research activities on genomics, on proteomics, in chemistry, and in bioinformatics. Okay? So all the research groups can benefit of these central facilities. So these central facilities run kind of service for internal users, the scientists, but also for external users, okay, via collaborative projects or outsourcing, okay. So all of our work starts really from a number of genome projects. We contributed to all the genome projects that I've indicated here. So we contributed sequencing and annotating and defining some of the plant, mainly plant genomes, and also some of the animal genomes. Some of those were already published. Some others are now submitted or under completion. So it's really from this kind of information that we start most of our activities. And I will give you 2 examples on how we use this information to really try to understand, as I guess you're doing here, some of the mechanisms, behind some of the behaviors. And we mainly focusing on diseases, sorting out and try to identify solutions for animal and plant diseases. So I just wanted to give you a little example of a project which is a European project called Macrosys. You can find information if you're interested on this project on the net. And so the scientist, my colleague Giulietta Minozzi, she's one of the heads of the animal genomics. She works on trying to understand the responses in cattle to this Mycobacterium avium, so paratuberculosis. Okay, so this what we call MAP, okay, causes this Jones disease. I don't know if you know this disease. Anyway, causes a number of, it affects the animal quite consistently. This Jones disease nowadays not treatable. Vaccine, they're not so efficacious, and it's a very costly disease for dairy production. And today the host genetic components, they have very low heritability. So our challenge was, and the still ongoing challenge is to identify some of the genetic components, behind the susceptibility to this disease, in trying to identify mechanisms of resistance. So the way we approached this one a couple of years ago was after the sequencing of the bovine genome, there were millions of SNPs which were identified, which were polymorphic in a certain population. So in collaboration with Illumina, we developed this 50K SNP chip. And what we did, we genotyped 500 animals which were cases of Jones disease and 500 controls. Okay, so then we phenotyped all of these animals for a number of traits relating to productivity, relating to immune response, relating also to their behavior. Okay. So the aim of this work was to really localize regions and genes which had an impact on Jones disease susceptibility. And also associated to that, once we would identify candidate genes, also identify the networks and the biological pathways, trying to then reconstruct and identify strategies to overcome this disease. So we used kind of standard methods nowadays. We carried out what we call the genome-wide association analysis. And we used genome-wide association using mixed models and regression. I guess some of the bioinformaticians, I don't know if they're here, but these languages should be quite straightforward for them. Maybe not so much for other type of scientists. And so we obtained some results that I will show you in a minute. And so the rationale was that we then identified areas which had significant association between specific phenotypes and specific genotypes. And then we looked at the genes which were within one megabase upstream and downstream of these regions, identifying at least clusters of 6 associated SNPs. Then we identify the putative function of these genes and trying to understand the network or pathways to which these genes belonged. So as you know, genome-wide analysis, association analysis is represented by these kind of plots. And so the significance of specific SNPs associated is given, by some biostatistical parameters. So those, it's a small set of SNPs which are tightly associated, okay, in these specific linkage groups. All of these are different linkage groups. Okay, so here are some of the tightly associated SNPs, which are candidate areas for us. So we, as I mentioned, we started to look at the genes subtending these areas, and we identified some of the genes which made some sense in our biological system. So all the genes in blue are actually in the areas where we identified significant association, and also they are following into this category. It's a general category: antimicrobial responses, but also small molecule biochemistry, cell morphology, and of course some aspects of cell death. So then we started to then develop what we call gene networks, and try to understand some of the genes which were putative genes involved in these responses, and how they were fitting in known signal transduction pathways or metabolic pathways. So we mainly worked on these antimicrobial responses, infection mechanisms, and cellular proliferation. And some of these data are actually coming from the human. Okay, so we try to translate from all the large body of human information back to the animal information. As we know, there are, of course, lots of similarities between the immune response in the two systems. So this work then, as I mentioned, was based on these 3 main approaches. And now we identified some of the regions, some of the candidate genes that we're doing fine mapping, trying to really narrow down to very specific genes and gene functions. And also alongside that, all of our putative genes, we use them in an in vitro cell system where we do kind of in vitro infection with a mycobacterium, and trying to really pinpoint the most important ones and their type of regulation. At the moment, we're looking at transcriptional regulation. Okay, so the second area that I wanted to talk is, of course, on rice genomics, which is the reason I'm also here collaborating with Vittorio. We're both working on the rice system. So we created 5 years ago this group, okay, and here I've just described our general vision, which is really what we aim in the long term to be able to do, and really is to support the breeding programs. So how plants will be best performing in the field, so producing lots of more rice with less diseases and more what we call eco-sustainable approach. Okay, so our mission is really to use the available information. Of course, we don't use any GMO because our institute is not really pro, as the whole Italian situation anyway. So we really try to use what is called biodiversity, so what is existing in nature, and really plugging in some of the molecular biology and genomics, which in rice are now very, very well developed, as now more than 50 genomes were fully sequenced of 50 different varieties. So we can use all of this information and exploit this information for our own interest, scientific interest. So here I would like first to acknowledge the scientists which were involved in the activities and the projects I will present to you. So our team is mainly of Italian scientists at the moment. If now we have a Chinese colleague, a senior scientist in China, which is going to coordinate some of the activities. So the project that I wanted to mention to you, it's called Rice Immunity, and really aims at studying the molecular mechanisms of resistance to a fungal pathogen, which is called Magnapothagresia. So this work is being carried out over the last 3 years in collaboration with a number of other institutions in Europe. So, so the problem is what we call the blast disease, okay? So the problem is really depicted in this slide. So 70% of the Italian varieties, so the rice which is grown in Italy, is highly susceptible to this fungal pathogen. And so the real phenotype that you see when the plant is attacked by this fungus, as the name says, you know, the fungus is really blasting out of the leaves, and then is really destroying the leaf completely. And I will explain you why this fungus works like that. So this one is a big problem at the really applied aspect, because Italy is the main producer of rice in Europe. Okay, well, Europe is not really producing lots of rice. We're producing just 0.5% of the worldwide production. But for Italy, as you know, risotto, you know, we have, you know, we're very much interested anyway. So anyway, so this one is the biggest problem in Italy. And in 2008, it destroyed 40% of the Italian production. So when it comes out, it's a major problem. So this fungus has a life cycle. So we can grow in vitro this fungus. So we know quite well. We already sequenced at least 10 or 12 different strains of the. So anyway, so there are spores which are transported by the wind, okay? And so they float in the air, and then they land on the leaf surface, and then they're able to germinate. But they just need some humidity, and then they'll germinate. And then they have a structure which is called a pressorium, okay? So they're able to really form a structure that really adhere to the leaf surface, so it would be very difficult. And then is really using brutal force, so it's osmotic pressure to really go through the epidermal, so the first layer, like our epidermis, okay? And then, so this one is called biotrophic phase, okay? So what happens is it gets into the cell, and then it doesn't want to kill the cell. It just wants to take all the nutrients, okay? So progressively, you know, it takes all the nutrients, and the plant is kind of starving, okay? It doesn't have any more nutrients. And then cells try to collapse, okay? And then at that stage, he thinks, well, okay, now it's better to destroy everything. Then he enters the necrotrophic phase, doesn't care anymore, okay? He says, oh, I have enough nutrients, I have enough spores, I've already replicated myself. So he starts to you know, it keeps going, and it keeps growing inside the cells. So it's an intracellular fungal pathogen. And then he bursts out, you know, of the leaf, and then he creates new spores, okay? So that's the reason when we will look at the phenotype, okay? So here are small islands, you know, full of spores, okay? So you see the center, and all around is yellow because the plant is deprived of all the nutrients. But then here in these areas, it activates also cell death mechanisms, you know, programmed cell death in the mechanisms. So the cells, you know, enter in a suicidal kind of program. So we wanted to really to understand, you know, some of the mechanisms behind, you know, these kind of events. And the angle that we wanted to take, so most of the scientific community started to understand and you know, to investigate really what we call host resistance. So really the strains which attack rice. So we decided a different approach. So we've taken strains which do not attack rice. They attack other cereals, you know, kind of wheat or sorghum, okay. So normally, and here is the example of this BR29, okay, rice is kind of immune, you know, to this. So we wanted to understand what really happens, you know, what kind of responses the plant activates. And then this one is the host pathogen, so okay, which is called FR13. So to this, the same variety, so the same plant is highly susceptible, okay? And then there are intermediate situations where we use other strains and we see a kind of intermediate response. Furthermore, there is also response which is genotype dependent. So that means individual 1, okay, is different from individual 2. And this one is for example, more susceptible, okay, to these strains, and this one is more resistant, okay? So, of course, as expected, they behave differently. So we think that, for example, this guy is kind of immunodepressed. He's feeling, well, this fungus, okay, let it go, and then who cares? So this one will have a very low fitness, okay? So for us to understand also the modulation of these responses is also very important. So we developed an in vitro controlled assay where we use these fungal spores, and within 5 days, we can replicate what really happens in the field. So we have methodologies where we infect in growth chambers these plants, and then we let it grow, and then we look at the phenotypes, look at the symptoms. So we can also take samples very early on after the inoculation. Okay, so within say 20 days, we can go from the seed to the result. Okay, but then the assay only lasts 5 days. Okay, so first we looked at a number of cellular responses. Here I've just put one example. Okay, so this one is kind of the spore which lands on the epidermal cell, which you see here. And then you have, at that stage, you can have different responses, okay? So normally you have no response. So the cell, okay, says, okay, you're there, I don't care. You're not really bothering me, okay? Or then the cell says, well, actually, this one is again the spore, and it tries to penetrate, and it says, well, actually, you're bothering me. And so I activate lots of cell death. Okay, then I stop you because you are a biotroph. You want to take all of my nutrients. So the plant says, well, I better stop you within that cell that you're trying to attack. So you're not attacking and not depriving the rest of the plant. Okay, so this one is a normal kind of response that we have in plants, but also in other animal and human systems. Okay. So in this case, okay, the kind of activation of cell death is more widespread, so it can spread to neighboring cells. And then in this case is the case of susceptibility. So it's kind of difficult to see, but anyway, this one is our spore. It actually penetrates the cell. This one is kind of the immunodepressed guy. Okay, so the spore and then the fungus penetrates and make this intracellular hyphae. Okay, so there is some local responses. Okay. But actually, the cell is still alive, okay? So eventually, these hyphae, well, they're just sucking all the nutrients, and then they will move out into other cells and continue, depriving from all the nutrients. So this one is what happens in susceptible plants, okay? And this one eventually causes the symptoms. So we can quantify, taking these 4 classes, okay, which were the responses to different strains, okay? And not surprisingly, we found that, for example, these strains, which is not adapted to rice, is adapted to other species, it actually, in most of the cells, okay, nothing happens. Okay, they're kind of type 1. Whilst, for example, the host, which, gives lesions, most is this type 4. Okay, so most of the cells are actually kind of susceptible to these to this strain. And then we have intermediate situations, okay? So we're very much interested in understanding, why then the plant, it actually in this case, stops at actually the ingress, the attack very early on, in such a way it cannot even penetrate the cell, okay? And actually there is a lot of very little cell death activation. While in other cases, actually, the attack of the fungus is blocked, but then there is a massive cell death activation. So we really wanted to understand some of the basis of the innate immunity responses. Okay, so a couple of years ago, we started, to do some Affy arrays. There were these 44K array, which is kind of comprising most of the genes which are expressed in rice. Rice actually has more genes expressed than humans, okay? So which is an interesting thing. But we have a lot of duplicated, large duplicated gene families, which also explains why the plants are able to withstand lots of stress, biotic stress, abiotic stress, because as you know, plants, they cannot move. But actually, they can move a little, but they cannot walk. They cannot hide. So they have to be there and able to respond to all sorts of different stimuli. Okay, so then we inoculated, okay, with our system. we did, of course, some biological replicates, and then we hybridized it to the Afi chip. Okay, so this one is our design. Okay, the design, as I mentioned, we have different strains. We used one specific type of plant, and then, of course, we checked the symptoms, and then we took RNA samples at very early stages because we wanted to see what early on happens, of course. And so then we did our microarray experiments. So here we've taken 3 biological replicates, what you see. So the mock inoculated, that means we just sprayed on the leaf, some gelatin or some water. And then those are, our 4 different strains. One which means it's fully susceptible, the plant. So the plant let the fungus grow inside, okay? The CL367 is actually another strain to which that specific plant responds activating cell death, okay? So it activates cell death and then is able to block the fungus before it gets infected the rest of the cells around. And then BR29 is actually that strains which blocks very early on, the ingress and the attack of the fungus. And BR32 is kind of intermediate. So that fits also on the whole transcriptome analysis, because as you can see, in this case, there is a big cluster of genes which are kind of activated and actually here downregulated, okay? So it's interesting because the cell really globally responds very differently, okay? And as you can see from this little clustering analysis, which was very reassuring for us, the 3 biological replicates actually stay together, because it was a bit worrying that because there is a lot of biological variability, if these replicates would not fit according to this scheme. Okay. So we're mainly interested on these responses because those are responses of the immune system. So the immune system of the plant responds very rapidly and then is able to block the fungus. Okay. So the other thing is that in these 2 cases, this one is a fully adapted rice strain and this one is not. Okay. However, these 2 are very tightly linked because both of them activate cell death. So at the end of the day, what we're interested in is, a cluster of genes, okay, which is specifically regulated, in these kind of events, okay, where the plant is able to block the fungus very early on, okay, or, a number of genes which have very different type of regulation in the different groups, okay. So we focused on some of these genes, trying to understand, their function. So the other interesting thing, there are in the database around 5,000 experiments using the same Afi array. So we could use all of this information also to study bioinformatically, okay, how genes were co-regulated. And I will show you one example on how we use this information. So of course, as all the people do in transcriptome analysis, we looked, overlaps and specificity of different types of interaction, huge number of gene lists, which doesn't really make any sense. Okay. So anyway, we started off anyway trying to understand the genes which were upregulated, downregulated in different types of interaction, and some of the genes were actually activated on only on some of these interactions. And I will show you just one example. So this is what we call non-host, which is the innate immunity response. And this one is the host response, so when actually the plant recognizes the fungus. Okay, so just here we did some, of course, gene ontology experiments, okay, and classification. And, I just pointed out, two interesting points. So here you see that practically in the non-host response, this class of genes, which are sulfur-containing defense compounds, that are completely absent, okay? So that means, in the host response, there is activation of a certain number of genes which we can identify as belonging to this group. On the other hand, you can see in the host responses we have another group of classes which are classes something to do with secondary metabolites, okay, which are actually they're not expressed or differentially expressed at all in the host. So those were entry points for us on trying to understand, which were, the metabolic pathways and the signal transduction pathways, which were activated according to one response or the other response. So of course, then we started using a software which places, all of our genes and gene functions inside metabolic pathways. Okay, so I just put one example. So here's secondary metabolites. Okay, so there are specific genes which actually are precursors of defense proteins. Okay, which are very well known, which are only activated in some interactions, whilst they're not activated in other cases. So likewise, we have cell wall modifications, biosynthetic genes, which are actually strongly downregulated in one situation and actually upregulated in the other. So we started then reconstructing inside the cell what was really happening, okay, and trying to really focus us on some metabolic pathways. And then we use knockout mutagenesis, site-directed mutagenesis on the plant, trying to understand how these gene family or gene families were actually affecting. And this one is some of the ongoing work. Okay, so of course, we cannot work on 44,000 genes. So we focused on some gene classes, and one of these is a class of transcriptional factors that we call worky genes. Okay, this one because there is this worky motif, okay, which provides specificity to the target sequence. So quite well-known transcriptional factor gene family. And actually, none of you, I think, knows this class of transcription factors because this one is a plant-specific transcriptional factor. Okay, so it's very interesting phylogenetically because it really developed only and it disappeared, in some other clades during evolution. Okay, so those are quite well-known transcriptional factors, and they're involved in a number of different responses inside of the plants to different stimuli. So this kind of gene family kind of exploded in plants. So there are more than 100 of these genes belonging to this gene family. So here we used two of the most important model species in the plant scientific community, and try to understand also how these genes were organized on the phylogenetic point of view. And so we found that most of these genes were actually belonging to mixed groups. Okay, so we could find them in these two very different model species, which are actually evolutionary, very distant one to the other. Okay, but there was a group which we call group 3C of genes which were only present in rice, and actually you can see that because of these two letters OS, whilst AT is the other model species. Okay, so we're very much interested because we thought that those adapted specifically to responses which were specific of rice, which were not happening in the other model crop. So we constructed a kind of whole gene family array. We really wanted to go into that because in the AFI array, not all of them were present, only something like 70% of all the genes. There are 104 genes in rice. So we looked at the expression of these genes in a number of different conditions, okay, of course, upon infection on the system that I just explained to you. And we started to then look, we were interested on genes which were co-regulated, okay, because we thought maybe those, of course, maybe they are involved, in the same pathways. And we find the genes which are modulated in specific conditions and they're co-regulated. Maybe those genes represent, complex, protein complexes at transcriptional regulation. So then we use all these AFI data to calculate the Pearson coefficient test, okay, which is a coefficient that explains how genes are co-regulated independently from from where they're expressed, and the kind of stimulus. It is really to say these genes are actually working together. Okay, this one is a working hypothesis. Okay, so all of these work is, as I showed you from the phylogenetic analysis, they're divided in different clades. Okay, so one of the findings is, as we might be expecting, there were early duplicated genes, okay, which are actually working together because those are early duplications, and then the function of those, remained, kind of the same. But if we look then at these two major groups we call coregulation 1 and coregulation 2, you can see that there are also a number of genes which are tightly coregulated. So the thickness of these lines shows you the level of coregulation. So the thicker is the line, the more coregulation we see on the system. Okay, so you can see that the same colors, this one was kind of expected. But there were tight correlations, say, for example, between these red genes and this pink gene, which was totally unexpected. So then we went on and we tested, functionally some of these interactions by two hybrid systems, okay? And we showed that some of these interactions were actually happening in vivo. And then according to that, we went into the in vivo system, and we knocked out, say, for example, this gene and this gene, and we found that knocking out only one of these genes, there was no phenotype. But knocking out both of them, there was a dramatic change on, for example, susceptibility to this fungal pathogen. So we see nowadays we really combine, bioinformatics information to gather, some leads, what we call leads, and then we go into the functional test, trying to really redirect as much as possible. So some conclusions. So we use whole genome expression data from these AFI experiments to try to identify signal transduction and metabolic pathways which were involved in innate immunity to different fungal strains. So we also used the whole genome transcriptome analysis of these transcriptional factors, and we found that there were specific patterns. We identified two main co-expression worker gene clusters, which were, as I mentioned, involving genes belonging to different phylogenetic clades. And now we're using this to functionally test the involvement of these work transcriptional factors. And so our aim is really to identify the master regulators of different cellular responses. So the one which are really up there and they regulate a number of responses. So if we really are able to modulate their expression, okay, we should be able to really have an effect on enhancing the resistance level of plants to these major fungal disease. So the reason I'm here is that Pietro and I are involved in a national project which is called ReasonNova. And we started some collaborative genomics efforts to unveil the mechanisms leading to the understanding of the interaction between rice and beneficial and pathogenic bacteria and fungi. So we're now moving into trying to understand not only a single fungus, how it interacts with the plant, but it interacts with the community, which we know is present on the leaf and is also present inside the plant. Okay. So this one is the system that we're using. Okay. So the plant can be grown in different growth conditions, okay, which are called wetland, okay, and upland. In a sense that here there is a lot of water and here there is little water. Okay. And we know that the communities change very much on the two growing habits. Okay, so we collected, a number of samples, from different parts of the plant, mainly focusing on the roots system because this one is the most important one for plant growth. And then, as we indicated here, we're taking in some of the metagenomics approach to trying to understand and pinpoint very important components of these microbial communities, and also trying to understand the interaction with pathogenic and beneficial microorganisms interacting with the rice plant. So, of course, a number of collaborative projects with different institutions in Italy and in Europe and also outside Europe. If you're interested to have more details, here's the website of our Genomics group. And I will stop here and take any questions. Thanks.